righty, I think we'll go ahead and get started here with our next Ivan NMR research workshop on industrial applications of Benchtop NMR. This time I'm going to pass it over to David Rice.
Hello, everyone, and welcome to our what we've decided is our 41st Ivan meeting online meeting since we started these just at the beginning of COVID. And anyway, I would like let me welcome everyone and let me turn things over to John.
Well, thank you, Dave, and good morning, everyone. Good afternoon, good evening, wherever you may be. I recall last March meeting, it was just turning springtime, and I think there were some folks with us from New Zealand and Australia, and I was sort of tongue-in-cheek joking around about we're just getting into spring, and well, now it's the other way around, so winter's coming up. Anyways, very good, very important subject material today on high resolution benchtop NMR. And just before the meeting started, we were just sort of joking around a bit about back in the good old days with Bruker WP60s and Varian T60s, and those 2.5 to 3 ton iron magnets would certainly not fit on a benchtop these days. But more, a little bit more importantly, with today's meeting, we're going to look at obviously applications and maybe sort of compare and contrast high resolution benchtop with high resolution high field, and do they augment each other? Does benchtop stand on its own merits, or maybe a little bit of both? But anyways, thank you for joining, everyone. And of course, MR Resources and Q1 Instruments, very, very happy to sponsor the Ivan meetings. As Dave mentioned, number 41 today. MR Resources have been around since 1985, and sort of everything NMR, if you will, sales of NMR spectrometers, magnets, consoles, service contracts, quench magnet recovery, moving and relocation, and so on. And in particular, they've got very, very good inventory of used and reconditioned systems on hand. They've got currently Bruker Ultra Shield, Bruker Avance 3 600s, Avance 3 700s, even Avance 3 950. Please do contact them for more information. The MR Resources sister company, Q1 Instruments, also happy to sponsor. And Don, would you like to give a few words on Q1, if you would, please?
Q1 is dedicated to developing NMR hardware and software to make routine NMR more accessible to a broad range of scientists and engineers. Our aim is to establish new and innovative products for demanding applications, complete 400 to 600 megahertz systems, as well as console replacements for older Bruker and Varian systems from 3 to 600 megahertz. So we all know that Q1 offers an excellent performance at these field strengths at an unbeatable price. Everyone knows that the probe is the key to performance, and Q1 offers smart tune and match probes made by Q1 Instruments. Our STM probes have a robust hybrid tuning mechanism with faster tuning, unmatched reliability, and improved throughput. A patented design, which is free from drift and hysteresis, offers excellent signal noise and superior solvent suppression. And it's made by the Q1 team in Zurich. The probe configurations are inverse, direct detect, and even fluorine on both channels. We also offer probe replacements for Bruker and Varian systems that integrate with Topspin and VNMRJ. And we also offer custom probes. This is a 4-channel probe for tritium investigation. And we also just delivered a low silicon 500 megahertz probe. We have a new accessory of a low temperature -20 chiller for VT, standard VT air. And we invite you to call us, write us, or get on the phone for more information. Thank you very much.
Thank you, Don, and very good, very capable product lineup. Please do get in touch with them. That said, Dan, I think you have a few words for us on spin sites today.
Yes, we support an Ivan Spin Sites forum that's currently on Slack, and Slack has changed their retention policy so that anything older than 90 days will be hidden. That basically accounts for about 99% of the content, the current content on Slack, making it much less useful. We've transferred all that content to a new platform called Zulip, which has agreed to host our forum for free. And in a week or two, we have currently 541 members. And in a week or two, you should receive an email from Zulip inviting you to join the new forum that will be hosted by their platform. That's it.
Thank you, Dan. And once again, hats off to you for all of the great work you're doing on those things. Krish, over to you. If you'd like to let us know about upcoming Ivan meetings and then to kick things off for us, please.
Thank you, John. Welcome, everyone. Just wanted to announce what is coming down the pike before I pass it to today's panel leader. In October of 27th, we have the next workshop that will be led by Tim Claridge of the University of Oxford on NMR super sequences for small molecule characterization. He'll be joined by Eric Skupche and Jonathan Young. And in November, Travis is coming back with the second team on part 2 of industrial application of Benchtop. It is becoming a very popular topic. That will be on November 10th, followed by on December 1st, Murali Nagarajan from Rutgers will do a panel discussion on introductory bio NMR use. So the more details of November, December workshop will be forthcoming. And that will end the 2022 series, and we will kickstart 2023 soon after that. Today, we have, as was initially introduced, Travis Gregar from 3M Corporation. He's the panel leader on industrial application of Benchtop, part 1. Take it away, Travis.
Great. Thank you so much, Krish. Want to certainly welcome everyone. Good morning, good afternoon, good evening. As you may have seen, I've given a talk a few times in this area, how I've used Benchtop NMR here at 3M and in the factories. And so I wanted to lead a few sessions with regard to other applications of Benchtop NMR, and some are rather unique, I think you'll see. We have 3 speakers today, 2 of which I was referred to by Nanalysis, just trying to reach out to different benchtop manufacturing companies to try to find speakers for this. And one of them is Anca Frinculescu. She is I've been asked to say she's not an NMR expert, but she is a user of the NMR as a pharmaceutical analyst at TICTAC Communications through the St. George's University of London, where she will be talking about 3,4-methylene dioxymethamphetamine quantification via benchtop proton NMR spectroscopy, method validation, and its application to ecstasy tablets. Can't say that very well. Tablets collected at music festivals. So I think this is a very different application. And just to remind you, because she's in London at this time, she will not be participating in the after hour sessions. And so if you have any questions, now's the time, or after her talk will be the time to ask. We can always follow up with questions in the chat at a later time. Anca, are you there?
Yes, I am. Thank you, Travis.
Great.
And thank you for reading the title. I don't have to do it now. It's good. So basically, as I was saying, I don't have to read the title. Again, thank you so much for doing that for me, Travis. I'm going to start with an introduction. So I'll talk a little bit about me, then I'll tell you a few words about TICTAC communications and what we're doing, and then focus the rest of my presentation on the MDMA data that we collected on the low field NMR instrument from Analysis. So basically, I'm a pharmacist. I have an MSc in forensics, and I work as a pharmaceutical analyst, as Travis mentioned, for TICTAC Communications. We are based in London, in St. George's University. And because I had too much free time, I decided it's really appropriate for me to do a PhD. So I'm doing my PhD part-time at King's College London. And as part of my PhD, I'm looking at portable instruments, or like small instruments that can be used for analysis, even on site. So far, I looked at 2 portable GCMS units. I did a little bit of work on the Radian ASAP from Waters, and now, what I'm going to tell you about today, it's the work I've done on the low field NMR, the 60 megahertz, and we did the MDMA quant on it. And this is done, but in the future, at some point when I'm going to have time, I want to look at some FTIR and Raman spectra and see if we can actually improve the results using machine learning, but that's for later on. Now, why? Well, I keep asking myself why, but the reason we decided it's important for people to know what's in their drug is because, first of all, it's very, very dangerous. It's probably fun for people that use them, but it can actually be very risky too. So we wanted to make sure that we can help them improve the techniques when they give advice. If they do the drug checking for people that use the drugs, it's important for them to know what they have to tailor the advice on. And then there are also like more worrying issues like overdosing, deaths, and probably the most worrying one is the fact that MDMA, which is methylene dioxymethamphetamine, is used by children. So as you can see, almost 4% of the 15-year-olds have used the drug. They know what it is. And then if we look on the other side, it's really expensive. It's expensive for medical, it's expensive for police, for border control. Like, to deal with this drug situation, it actually takes lots of money. And you can see like public health in England. I know this is a very outdated number, but I couldn't find something more recent. But even in 2010, 2011, they were spending 10.7 billion pounds on illicit drugs, which is quite a lot. And as I said, it's really, really important in order to avoid all these costs, in order to avoid loss of life. It's important for people to actually be able to monitor the drug market, and this can only be done by analyzing the drugs properly and accurately. Now, back to TICTAC Communications. We are a home office licensed company specialized in drug analysis, drug identification, and drug information, but lots of times we are mistaken for the confectionery brand, and I don't blame them. The name is very, very similar. And well, our name means something else, but I'm not going to bore you with the abbreviation for that we use to actually come up with the name. But what's important is like people actually think we are the confectionery brand. And that was really funny because this guy actually made my day. He sent us a letter complaining that he only got 96 TICTACs in his container. And Trevor actually wanted to send him a full box again. But as we said, we are not a confectionery brand. It wasn't our problem, but it's still nice to hear from these people. So there are actually some things that we have in common, because you can see there are some MDMA tablets that actually have the TICTAC logo, and then the small containers, I'm not sure if you can see them with the orange background in the bottom, in the center, they contain cannabis buds, and it's a very, very small container. So people actually took the struggle to put the cannabis buds inside those containers. So as I said, we do have some connections with the confectionery brand, but we are not the confectionery brand. What we actually do, we collect drugs, lots of drugs. We collect both pharmaceutical and controlled drugs. And I don't want to brag about it, but so far we collected more than 37,700 products with over 4,400 ingredients from 1,200 suppliers, and we have lots of pictures. And the reason we collect them is because we need to populate our drug identification software. We need to update the FTIR and Raman spectral libraries. We use them for drug analysis. Drug images are used by different clients in medicine management systems or robotic dispensing. And also we use the large collection of drugs to assess different drug analysis instruments and techniques. And today I just explain you a little bit. I mean, you'll kind of see a little bit of the drug analysis and the assessment. Now, the fun part of my job is that I actually go to 2 music festivals in the UK. So we do the analysis on site already, and it's definitely more than music to us. Basically, when I give the presentation and I talk about the amnesty bin drug analysis, I ask people if they know what it is, but that's not the case now, so I'll just explain it. So really sorry if there are people in the audience that already know what it means. I'm just going to bore you with the details. So basically in the UK, when you go to a music, and probably in the US too, when you go to a music festival, you have to pass through security, and just before that area, you have the amnesty zone, where if you have any drugs on you and you all of a sudden get scared because you can see the security team with dogs, or I don't know, you can see other people being searched, this is the time when you can actually give your drugs away and nothing happens to you. So basically they are, you can put them in one of these amnesty boxes, and that's it, you can still go and enjoy the festival. Now, if you were brave enough to actually get to the security area and you have drugs on you, you are being searched, because sometimes, like, you know, it depends, like the search is, it depends on the weather, it depends on the number of people, so the length of the queue. Sometimes they have a step, so they have to search 1 in 3, 1 in 5, 1 in 10, again, dependent on various factors. But if you are searched and they find something on you, they're going to take it away. If it's personal use, they're going to take it away, put it in the same amnesty box, and then you can still go and enjoy the festival. Now, if you have too much, I mean, if you have more than personal use, they're going to call the police. And this is what stops, like, for us, we're not going to see those drugs anymore. We only get to see and analyze the drugs that have been given away by festival goers or seized by the security team. So a little bit of what we're doing on site. Before 2019, we used to take some FTIR and Raman instruments to use on site. We do the visual ID on the solid dose drugs, so for tablets and capsules, and then we bring all the inconclusives back to the lab and we run them on the GCMS and also the new tablets. But there are limitations when we are on site and we use the FTIRs and the Ramans. There are limitations, and these are just 2 examples. So basically, we came across these clear capsules that contain a brownish powder. We ran them on one of the Ramans, it came back as inconclusive. The other one was, again, a very noisy spectrum, so inconclusive. And then the FTIR said it's modified starch, which, if you think about the fact that the active ingredient that was detected on the GCMS was 4-hydroxymethyletryptamine, it actually makes sense. It probably was modified starch because that's a potent drug, so it was just a tiny amount of the active ingredient in the powder. But again, that wouldn't help the harm reduction team. And then this purple minion tablet, actually, I was a bit cheeky with this one. I first ran it on the GCMS, and because I don't like doing tablets on the, I never do tablets actually on the FTIR and the Raman instruments, I decided to run it anyway, just to show that they are not working. So basically, this was N-etylpentilone, and I'm not sure if it happened in the US too, but in the UK, one summer, we had quite a few cases, like hospitalizations because of this one, which was missold. It was actually, people were expecting to take MDMA, but they actually got N-etylpentilone, and the side effects are a bit AG and a bit different than the MDMA one. Now, the first Raman said it was inconclusive. The second one was actually a bit better. So we kind of guessed the group. It was still MDPBP. It's a catenon. It's a synthetic catenon. So it kind of got the class, but it wasn't the correct one. It was a different one. And then the FTIR said it's a Parexil, which I think is like toothpaste or something, but that usually happens. I usually get that hit when I have to run MDMA tablets just to show that it's not working, of course. On the FTIR, I usually get Parexil, so maybe it's one of the excipients, I have no idea, but yeah. And now, in 2019, we decided we can do better. So we still took the FTIRs, we still took the Ramans, but we also took the 2 portable GCMSs that I'm looking at for my PhD, and that was actually quite helpful. So we managed to run some of the inconclusive on site, we managed to run some of the new tablets, but as you can imagine, we are there just like 4 to 5 days, and we get lots of samples. So even separating them and bagging them, it takes like really a big amount of time. So we do not actually have the time to run everything on site. We still have to bring them back to the lab to run them on the GCMS. And you have to trust me, we don't have the time to run them all, because these are just a few of the new tablets that we got in 2017. So I don't know, this is also to show you how it actually evolved. The ecstasy tablets and the whole market is like really developed now. If you remember the very beginning of MDMA on the drugs, on the dance scene, it was just a white round tablet with a logo on one side. Now they actually are very bright and colorful. They take the shape of the logo, and you can see some of them are even by color. So the bottom right on this slide. Unfortunately, I don't have any updated pictures, so we don't have any 2019 and 2021 and 22. We do have lots of tablets, but I did not have the time to actually put them together on a slide. But they are, even these ones, 2017 and 2018, I think they are quite interesting. And yes, we run them on the GCMS, then we photograph them, we add them to the database, but we realized knowing it's MDMA is not enough. So basically it's important for people to know how much MDMA is in the tablet, although this is really difficult to actually use in harm reduction, because you won't be able to analyze the tablet that's going to be taken by the festival goers. But I don't know, at least it gives you an idea, you keep up with the trends. And in order to do that, we decided the GCMS, we could have used the GCMS, but it would have been a little bit more expensive and then maybe time consuming too. So we came up with this method using the low field NMR from analysis. It's the 60 megahertz, of course, and then we decided to use the internal calibrant method, and for that one we chose ethylene carbonate. And after we developed and validated the method, we wanted to make sure that it actually works, so we tested the diagnostic ability on 100 tablets that we collected at music festivals. And these are just a few, I mean, not a few, quite a few samples that we run on the NMR already. Now, for the sample preparation, I'm not going to bore you with that. It was quite easy and straightforward. The only thing probably was that we had to, instead of dissolving it in 0.6 and transfer the solution, because in tablets, some of the excipients are not soluble in the MSOD6. We had to actually add 1 ml just to make sure we don't transfer any of the solids. And then for the experiment parameters, I'm not going to get into that because you probably know way better about this than me. So if you have any questions on that, Alex is here and he's going to be able to answer all of your questions at the end or even later on. Now, I just wanted to show you, because basically we chose ethylene carbonate. It took us a while to actually come up with this one. We gave it a go with DNB before, dinitrobenzene. It didn't work. And then when we swapped to this one, it was because, first of all, it's soluble in the MSOD6, which is really important. Then it's inert, so it doesn't interact with anything in the samples, not the excipients nor the MDMA, the active ingredient. And then it's also important to make sure that the peak for your internal calibrant, of course, doesn't overlap with the peak of interest. In MDMA, we chose the methylene from methylene dioxide, so you can see it's around 6 ppm, which is fine because ethylene carbonate was 4.5 ppm. Now, if you look at the spectrum in the middle, so the second one where we have ethylene carbonate with MDMA, the MSOD6, you can see around 9 ppm, there's a bump, a broad signal there, which was for the nitrogen attached protons. So in order to remove that and be on the safe side, because that apparently can show up in other areas of the spectrum, we just added water to the sample prep and everything was fine. You can see there are no broad peaks after the D2O addition. Now, to validate the method, we followed the UNODC recommendations. So basically, we started with the external quality control. We had 10 of the samples analyzed by a different lab, and then we compared the results. And as you can see in the table here on the right, the deviation was minimal. And then in the middle, you have a snip of the spreadsheet. So basically, again, if you look at the calculated purity on the low field NMR, it was 0.4113. while the LCMS, the percentage was 41.4, so basically similar. Now, to test the selectivity and specificity, we looked at quite a few cutting agents and excipients. So let's start with the cutting agents. We did all of them, like levamisole, lidocaine, phenacetin, benzocaine, caffeine. And you can see, I mean, maybe there was a small issue with benzocaine because the labile proton is around 6 ppm there. But of course, if you add the D2O, that's going to probably go away. Luckily, we didn't have to worry about that because we didn't have any benzocaine in our samples. So basically, prior to the analysis, the quant analysis, we ran everything on the GCMS for the qualitative analysis. So already knew what to expect in those tablets. So everything was fine on this side. And then for the excipients, we ran quite a few of them: glucose, sucrose, mannitol, sorbitol, you name it. Lots of them, but the only one that I wanted to show you today was the lactose, because that's the most common one used in MDMA tablets. So, and you can see there was around 4.5 where we have our ethylene carbonate. There was an interference first, but then when we started adding water, that disappeared. So basically, we were safe. There were no problems after that. And then for the compounds that have a similar structure, the only problem was MDA, which is methylene dioxyethyl amphetamine, and MDA, methylene dioxyamphetamine. The thing with these ones was that we only had the mixture, so we only had street samples, and this came mixed with MDMA already. So it was really difficult for us to distinguish the two drugs. You can see there are some slight differences in the spectrum, but for the methylene dioxy peak, we don't know if it's just the MDMA or if we're integrating both of them, which is probably more likely. So when it comes to this, you have to actually do further analysis. But everything else was fine. Then the method was linear in the working range. The limit of quantification was 0.33 milligrams per mil, and then the limit of detection was 0.1 milligrams per mil. Then to test the precision, we ran the same samples in the morning and in the afternoon for 5 days in a row. And you can see again the deviation is very, very small, and it's well within the UNODC recommendations. We tested recovery as well, because we have a crystal sample that we actually quantified on high field, which is the one that we used throughout the validation anyway. So we knew how much we were adding into our mixture, and we basically knew what to expect, what the results should look like when we ran them on the low field NMR. I couldn't actually press the tablets. I wish I were able to, but I did not press the tablets. I only had the powders, and then the excipients. They are commercially available. You can buy them online, and you have everything in a bag, everything that you need, like even the dye. It's all there in the same mix. And again, for these ones, the results were fine as well. And then getting to the testing batch, there were 100 tablets in total, but we split this in 5 groups. So we looked at some very, very small concentrations of MDMA, because we had those quantified in a previous project. So we wanted to make sure that the method and the instrument will work on those too. Then we chose some tablets that contain some other active ingredients, like some of the cutting agents or similarly in structure products. And then for the within batch variation, we took only 2 samples, but for each one of them we had 10 tablets. So basically those samples were submitted. It wasn't actually one of the amnesty bean samples. This was a police seizure, but it was already non-evidential. So there were quite a few tablets in those 2 samples, so we could actually choose 10. And we know for sure they were seized from the same persons, like two persons. And then we looked at some 2019 and 2021 tablets, and that was mainly because we already published a paper with the MDMA quantum dissolution for tablets that were seized between 2001 and 2018. So we just wanted to kind of continue that project and see the changes. Now, As I said, low dose and cut tablets were fine. Nothing else to say on this one. I just want to show you the within batch variation because this is quite interesting. So if you look on the left, we have the Red Bull, the pink tablet, and you can see the variation of MDMA. It's minimal in that graph, so that was fine. And then the difference from the mean and the maximum is only 14%. When you look on the right side, when you look at the trophy tablet, that's actually quite tricky because you can see the variation is quite high. So basically you have the lowest concentration, the lowest amount of MDMA, sorry, was roughly 73 milligrams, and then the highest one was 120. So that's 50 milligrams in between the doses. And it's really important because if people start with the low one, let's say, so if they take the 70 something tablet, maybe they expect, maybe I don't know however they've taken MDMA before or what they expect from these tablets, but if they expect the effects to be stronger, and this is not the case when they only have 70 milligrams, let's say, they're going to take another one. And what if the other one that they're going to take is the 120? That's already too much in the system. So that's a bit problematic. And then to show you the comparison, so the 2019 and 2021 results, you can see it went up from 2018. That was quite a big increase to 2019. Now it's going down, but of course not to the same level from before 2019. So it's really important to actually monitor the trends and keep up with the trends. And of course, we didn't get any samples in 2020 because everything was shut down. But we're hoping to get back on track now. And just to conclude, the method was successfully developed and validated using the 60 megahertz from megahertz benchtop NMR from analysis. We used the internal calibrant, everything was fine. The specificity, selectivity, precision, accuracy, and recovery were tested, and the results fell well within the UNODC recommended limits. When we ran the testing batch, the 100 samples, we observed that the low doses and the cut tablets are fine. They need to be a bit careful with the within batch variation. And then it was really interesting to see that there was an increase in those for the 2019 and 2021, still higher than 2018 tablets, which highlights the importance of monitoring the drug market. And yes, basically the Benchtop NMR was efficient and sufficient in doing the MDMA quant in ecstasy tablets that were collected at music festivals. And if you want to know more about the method and the results, you can actually read our paper. It was published a few months ago. It caused quite a stir, but what can I say? We're dealing with that now, and I take it as a good sign. And I just want to thank you for your attention, and I want to thank all of the people that worked on this project with me: Alex, Juan, Susie, Nanda, Vincenzo, John, and Trevor. And again, thank you for your attention. And
Excellent.
as Travis said, I'm going to take some questions now, just the nice ones. Keep the hard ones for Alex at the end.
Thank you, Anca. Very, very interesting. Who knew that? I mean, NMR can be used for a lot of things. A question that I had is, are you in a regulated lab where you had to qualify the instruments?
Yeah, well, we are not an accredited lab, if this is what you mean. We don't do everything that we do here is non-evidential. And the reason for that is because I'm the only analyst. So I could not be in the lab and also in port. So it's not something that we are interested in.
Okay. Thank you. Thank you.
No worries.
Any other questions?
I'll be happy
Jose.
to. Yeah, Anca, very nice presentation. Very
Thank you,
interesting
Jose.
application. You mentioned on the initial slides fluorine NMR. How often do you run into fluorinated analogs?
We do get quite a few when it comes to synthetic cannabinoids, but we don't see those at festivals. And I've done one actually for the master's. My final project was to look at 2 synthetic cannabinoids, and I just monitor the concentration in different smoking mixtures, but I've done that on the GCMS. So maybe it would be helpful in the future, but no, I haven't done anything on that so far.
Thank you.
Thank you.
So, hi, Anca. This was really nice talk. So I've been working forensics for many years in a different lifetime almost. We use normally several different methods, right, that you always need to use to validate a type of drug. Do you think that the Benchtop NMR has sufficient specificity to use as a complementary to, for example, a chemical color change that we would normally use to screen first for that drug, which would open its use for application in forensic testing labs, and getting that UCAS accreditation for those labs that do use Benchtop NMRs?
I'm going to say it's a good question, but I'm going to ask Alex to answer to you because I really don't know. As I said, I'm not an NMR expert, and this was the first project that I actually did on the instrument. So maybe after everything, when you have the discussion, Alex from the analysis is here, so he's going to answer that question, I promise you.
Thank you. Good question.
Interesting question. I could also jump in right now. So thanks for asking. So if I understand correctly, you're asking for if it's a screening method possibility on Benchtop NMR as well. So we are looking into and developing an automated kind of detection for compounds for illicit drugs, and kind of a library search that would be able to accomplish that. And as you say, like as an orthogonal method for sure, but also as an independent method that give you qualitative and quantitative information. I hope that answers this question, but we can also discuss this further. Thanks.
Thanks, Alex. Any more questions for Anca?
Yes, one more quick one. Why DMSO and not just D2O?
We tried quite a few. When I started, I tried quite a few of them, and we decided that was the best solubility. Alex is nodding, so yes, I'm guessing this was the right answer.
I mean, there's in theory, there could be also some unconverted free base in the sample, and that would not dissolve in the D2O. So then you would have to add some HCl, I guess. So the sample prep would be a little bit more tedious. With the DMSO also, it's interesting. You get more information on the matrix of the sample, I think. But of course, more dissolved analytes gives you a more crowded spectrum as well. So if it becomes too crowded, then you might decide to also look at a D2O spectrum.
Thank you. Thank you. Any more last minute questions for Anca? It's getting late where she is. All right. Well, thank you so much, Anca. Very interesting talk, and certainly, I think certainly opened my eyes into different areas, and likewise for the rest of the audience. So thank you very much. Because we spent a little more time with Anca in terms of questions, the talks will run a little bit later than normal for Brenna and Jose. But so I just wanted to give everyone a heads up. The talks will be going a little bit longer prior to open discussion. So thank you. Have a
Thank you for inviting me, Travis. Thank you all. Bye.
Bye-bye. So after Anca, we're going to have Brenna Brown. She is a National Laboratory Service Manager at Brenntag, where she will be talking about the application of Benchtop NMR for investigating the performance of H2S scavengers. So welcome, Brenna.
All right, well, thank you for the introduction, Travis. As Travis mentioned, I am with Brenntag Canada. I'm located in Calgary, Alberta, which is the traditional lands of the Blackfoot Confederacy, the Tsuut'ina Nation, the Stoney Nakoda Nation, and Métis Nation Region 3, which we acknowledge in the spirit of reconciliation. What I am talking about today is work that I've done on using Benchtop NMR for H2S scavengers. So what I'm going to go over today is I'm going to begin with an introduction to H2S scavengers. What are some of the challenges with H2S scavengers? We're going to talk about monitoring scavenger reactions, and then we're going to talk about why Benchtop NMR is just so great, which I think this is the perfect audience for me to get on my soapbox and say, Benchtop NMR is amazing. I will show you the work that I've done in applying Benchtop NMR to scavengers. We're going to look at proton and carbon NMR on samples from the field, as well as some laboratory experiments that we did in reaction monitoring. And I will end with some conclusions and some future work. The work I'm going to talk about today has been published in Magnetic Resonance in Chemistry in 2020. The references throughout, so you can check that out if you are really interested in it. All right. What is a scavenger? Why do we care about scavengers? Well, it has to do with sour gas. This is produced from the breakdown of organic material, and it is highly toxic. It is considered immediately dangerous to life and health at concentrations above 100 parts per million. On top of that, it is corrosive. So it is really hard on all of our infrastructure. And 1/3 of all natural gas in Alberta is sour. We use H2S scavengers to remove that sour gas, and non-regenerative scavengers irreversibly bind H2S. And we're talking about irreversible scavengers today. The most common irreversible scavenger is 1,3,5-tris-hydroxyethyl hexahydro-S-triazine. Now, that's a bit of a mouthful. So for the rest of today, I'm just going to refer to this as triazine, and that is compound 1 located in the screen here. It is a non-regenerative scavenger, and it can react with 2 to 4 moles of H2S. So the reaction scheme is shown here. You start with triazine. It reacts with 1 mole of H2S to produce this intermediate 2-thiodiazine and a mole of monoethanolamine, and it reacts with a 2nd mole of H2S to produce this compound here, which is dithiazine, and a 2nd mole of MEA. Now, those 2 moles of MEA can potentially also react with H2S, which is another scavenger. This reaction is dependent upon the amount of carbon dioxide. If we have a lot of carbon dioxide in our gas stream, then this reaction doesn't take place. And that's why we say it can be 2 to 4 moles depending on your concentration of gas. What is the main issue with triazine? Well, overspent scavenger. So when we use our non-regenerative scavenger, we call that spent. If we use it too much and we have a lot of this dithiazine product, it creates this solid scale. So it'll start to react with itself, and it forms this amorphous solid material, which you can see in this picture here. And that causes all kinds of equipment issues and downstream problems. You definitely don't want any of this getting into the gas that you are producing. So operators at gas processing plants are going to try and prevent the formation of the solid product while still maximizing their scavenging capacity. And that is one of the big challenges that we're talking about. So how do you monitor the reaction? That's the question before us. There's, of course, GCMS. We see GCMS all the time. Now, GCMS is great for low-level quantitation, but it's destructive technique. There's a lot to do with sample prep. It takes a while. The runtime is about 20 minutes per sample. And your molecules have to be volatile. And not all of these scavengers have volatile molecules. So that's a real issue. We've seen some work with Raman spectroscopy. And Raman is great for the fact that it can do aqueous samples. Most of these scavengers are in water, so that's really handy. It's a non-destructive technique, but the molecules have to be polarizable. It is not as sensitive, and it's looking at bond types rather than actual molecules. And if we compare our thiodiazine to our dithiazine, well, those bond types are pretty similar, and there's a lot of issue determining the difference between them. Our operators are seeking to prevent the formation of dithiazine. They need to be able to tell the difference between these two molecules. So that is where NMR comes in. What is awesome about NMR? Well, it is fast. You know, a proton NMR, you can get that done in a few minutes. It is great. Little to no sample prep. These things, add a little bit of deuterated solvent, good to go. It's non-destructive. One of my favorite things about NMR is the fact that it is inherently quantitative. We use it for all these qualitative applications, but oh, lo and behold, it's also quantitative. How amazing. You get detailed molecular structural information. You can use aqueous samples. And then for me, triazines are well characterized in the literature. It makes my job a lot easier if we already know what the NMR spectra look like. Problem with NMR is those very large, expensive systems. And this is something that in industry, it's really difficult to try and go to Brenntag and say, you know what, you should buy me this $1 million instrument because it's just so fantastic. On top of that, you have the high stray fields. It requires the cryogenic liquids. And in the time of a helium shortage, that's a problem. And of course, it's not as sensitive as GC. Well, one of the things that I love is that high performance benchtop NMR overcomes so many of these challenges. Look at how cute and little that instrument is. That is my benchtop NMR, and right behind it is an ICP OES. So you can see just, it does not take up a huge footprint in your room, in your laboratory space, and it doesn't require cryogenic liquids. It isn't that expensive. So here we get to bring in the power of NMR into our systems. And this is what I absolutely love, because to me, NMR spectroscopy is the best way to characterize any reaction. All right, how do we apply this then to NMR triazines? Okay, well, what we did was we had 4 samples of spent scavenger from field. So this is from gas processing plants who have used our H2S scavengers, and they're sending us the spent samples to analyze. So we had 3 samples that came to us from supplier A and 1 from a different supplier. So they all started with the same MEA triazine, but from 2 different sources. It was the same active triazine, the same concentration, and reasonably similar field conditions. So we expect that these should all look the same. To prep the sample, I took 300 microliters of sample and dissolved that in 300 microliters of deuterated methanol. And we ran it on the Analysis NMR Ready 60 Pro instrument. Here are the conditions on the field. So you can see 64 scans, 1 second delay. It takes about 4 minutes to acquire the proton NMR, so really fast. Carbon NMR, carbon already takes a long time, so we ran it for about 16 hours to acquire. As you'll see, we get pretty good carbon NMR, even running it at 15 megahertz. And what does the NMR spectra look like? Well, here is the NMR spectra, both proton and carbon, of unspent triazine. So pure compound, hasn't been reacted with H2S. What we see here are 3 distinct peaks. We see the triplet, singlet. The singlet is the methylene protons between our 2 nitrogen atoms. And then we see this singlet here, or excuse me, this triplet here, which corresponds to these protons adjacent on the MEA side chain. Carbon NMR, again, we see the 3 carbon moieties that we expect. This looks pretty good. So let's take a look at the NMR spectrum of a spent scavenger. This is a lot more complicated. So starting on our left-hand side, the first thing that we see are 2 singlets. These 2 singlets correspond to the methylene protons on the ring of dithiazine. Then we have 2 more singlets. These 2 singlets correspond to the methylene protons on the intermediate thiadiazine. And right over here, this particular triplet is very interesting because that corresponds to this methylene proton here on the starting triazine. So keep in mind, these are spent scavengers. So they're not scavenging H2S anymore. And yet we still see unspent triazine in here. In the middle here, we have peaks I've labeled 3E and 3F. This Michigas refers to all of these other methylene protons throughout these rings that all overlap. And there's not a lot of information to be gained from there. But most of what we're interested in is this peak 3G, because we're really expecting that there's not going to be any unspent triazine. It should be gone. So to confirm, in fact, that we do see unspent triazine in these field samples, I did a series of spike experiments. So we took the sample of the spent scavenger and spiked it with 100 microliters of unreacted triazine and compared relative integration ratios. If the peak G is related to unspent triazine, we expect that to be the only peak that changes in integration ratios, as well as peak E, where the other protons for triazine are. And that's exactly what we saw. Here are 3 samples we spiked, and in every case, we see that the increase in peak G occurs, which tells us, yes, there is unspent triazine in these field samples. Okay, right there. So taking a look at the carbon spectrum. What do we see in the carbon spectrum of spent triazine? As you can see, there's a lot more peaks in it. What I love about carbon, though, is usually you don't have that overlap of peaks. So when we see something like these triazines, dithiazines, where there's a lot of overlapping proton peaks, carbon can give us some resolution. There was a lot of peaks here, but going across, I can find the carbon peaks that correspond to my unspent triazine, which again confirms that yes, we do have unspent triazine in this reaction mixture. Then we have the peaks that correspond to this intermediate thiodiazine. And we have the peaks that correspond to the end product, the dithiazine. We also see the peaks that correspond to the NEA that is produced as a byproduct. So we are able to see all of those peaks as we move through the carbon NMR. The question now is, can we see how this progresses? So we see that we have this mixture in our spent samples of triazine, dithiazine, thiodiazine. Well, how does that change as we react with H2S? This is where we set up an experiment in the laboratory to determine that. We took H2S scavenger and we put it in a tube. Then we passed a mixture of H2S, CO2, and nitrogen through it, and we detected the gas coming out using GC. Once we start to detect H2S by GC, we call this breakthrough time. This means that our scavenger is 100% spent. It's not scavenging H2S anymore. And we were able to adjust the time that we ran the experiment from 100% to 80%, 50%, and looked at those samples via NMR. Just a moment for safety for you. We are running an experiment with compressed H2S. As I said, this is a highly toxic gas, so it is important that we take some important safety controls. Engineering controls, we have a separated H2S lab. This is where we house all of our H2S with specialty regulators and a specialty manifold completely designed to work with H2S gas. And we have specialty fume hoods that have scrubbers on top of them to remove any H2S in case there is a release. For administrative controls, we have alarm systems. So we have special H2S alarm systems. We wear personal monitors for H2S when we are doing this experiments. We have SOPs, we have training, we have signage. We let the entire site know that we are doing this. We make sure there's no one on the roof. And then for PPE, we have our flame retardant lab coats, safety glasses, gloves, and we have SCBAs as well available as well. So self-contained breathing apparatuses. All right, we're good to go. We got safety handled. It's now time to do the experiment. And here's what we noticed immediately was there was a shift. And that is because the lab samples had a pH of 8.9. And of course, pH impacts chemical shift. So processing these samples, we did have to adjust to the pH to 10 using a 2.5 molar solution of sodium carbonate. You can see the NMR there and the impact of pH on chemical shift. All right, here is what the reactions look like from 100%, 80%, 50%. What I find particularly interesting when we look at these data is that you see very, very little of the dithiazine up until 100% spent. You don't see very much of it at 50%. We start to see it increase a little at 80%, so 80%, and we still see very little of the dithiazine. And then once we get to 100%, that is where we see it. But looking at the relative integration ratios, it still seems that thiodiazine is the dominating species. This is particularly interesting because if you look in the literature for information on thiodiazine, there was this understanding that it didn't stick around, that reactions went immediately from triazine to dithiazine. That was it, that intermediate two just didn't stick around. And really that's because it's not stable if you go to put this in a GC and you can't differentiate it by Raman. With NMR, we were actually able to separate that out and see, oh no, that thiodiazine is there and is playing an important role. So if we pull all these data together, what we can see by NMR is there is still that clear progression from triazine to thiodiazine to dithiazine. If we go through the data from lab samples and field samples, the spectra are nearly identical. So again, that shows that this is monitoring quite well and that the performance in what we see in the lab when we design H2S scavengers does accurately reflect what is going on in field conditions. The field samples from different suppliers also produced the identical spectra. So if you are purchasing that starting triazine from Brenntag, or you're purchasing it from a different supplier, the reaction is still going to be the same. What we did learn as well is that some triazine still remains after breakthrough. So the reaction conditions do not push this to 100% completion. Thiodiazine is a reasonably stable intermediate, and we do see that dithiazine only builds up once you're over 80% spent. Taking a look at conclusions from this is NMR is a valuable tool for H2S scavenger analysis. Both carbon and proton are valuable. What we're working on now is developing some quantitative NMR methods. And as we saw from Anca right before this talk, there's a lot of power in using NMR for quantitative methods. Other areas that we apply NMR is reaction chemistry, product evaluation, purity evaluation, and product quality determination. That is huge for me in Brenntag because I'm also responsible for the quality control. And when you get rail cars of solvents and you need to make sure that you're sending out correctly what the material is, NMR gives us a really fast, really specific information that just isn't matched by other types of quantitation and qualitative analyses. So those are all areas where we like to apply NMR within Brenntag. And finally, a few acknowledgements. So Dr. Elena Tranina is the one that provided me with laboratory samples. She's now retired, but she was a great help. I have this fabulous team of chemists that I have the great pleasure and privilege of working with and leading at the Calgary Technology Center. And we have this collection of folks here, Ryan, Morgan, and Matt, who diligently went out into our field and worked with our customers to collect the samples that we used in this presentation. And with that, I am happy to answer your questions. Thank you.
Very nice job, Brenna. Thank you. Very nice job. Questions? I've got it. Is your starting triazine a solid or a liquid?
It's a liquid.
Okay. I'm just wondering, as it precipitates, so your first reaction with H2S, is it still that intermediate a solid or a liquid?
It's a liquid, and the N-dithiazine is also a liquid. It's not until it starts to react upon itself that then it starts to form a solid scale. And it's not clear exactly when that happens. It just does seem that it's once you have overspent it. And it's unclear yet. I don't know if anybody has done these of like, does it just have to be time if you leave it in your reaction chamber for a while? What frequently happens, because it is expensive to purchase these regenerative or non-regenerative scavengers, is in the field, they'll just top up their tanks. So they'll take some out and top it up with fresh scavenger. They won't necessarily replace all of the scavenger. So you could imagine a situation where you have some unspent scavenger, fresh, but you have overspent scavenger that's still sitting there and collecting and reacting further.
I'm wondering if the density of that precipitate, becomes a little bit more porous, that, if you had a way of compressing that precipitate in a way to help densify, to give it a better way for NEHS, to use up more of that starting triazine.
I don't think anybody's looked at that. That would be really interesting to see.
There you go. Until next time. See who else? Dave, do you have a question?
Yeah, Brenna, really nice, really quick. That was a challenging study. I'm impressed. You're always going to struggle with trying to get your integrations on those types of spectra when the molecules are very similar. And 2D NMR is fraught with peril to do quantitation. But I look at that and think quantitative HSQC may be valuable. Have you looked at that at all?
No, but I want to. So these are all on my very much to-do list. I would love to. That sounds amazing.
Very nice. Excellent. Good question, Dave. Anyone else? Well, hopefully you'll be able to stick around for a little bit, Brenna, after the second talk. A question from Michael came in. Is there a way to dissolve the solid overspent scavenger and run the NMR?
Yes, we can explore dissolving it in DMSO or in chloroform. You're able to dissolve the solid in other things. We've done a little bit of that where folks were sending us in solid samples. They didn't really get into the solid samples. It's unpleasant to work with the solid scale because it has an odor similar to a skunk that has been run over and left to rot in the sunshine for 5 days. But, it is doable.
Good analogy, Brenna. Krish, did you have a question?
Just wanted to, I'm actually pretty pleasantly surprised that you're watching my mute off. Yeah, I am actually interested to understand a little bit about your, when you say quantitation, following up on Dave's comment, quantitation by 2D, I am still wondering, you are still looking at relative change in whatever the amplitude or volume integral, or not absolute quantitative number. Is that correct? Is understanding? In that case, if that is the case, let me finish my statement before I pass on this. If that's the case, in my hands, just repeating to these faster, like SFHMQCs or SFHSQCs type of experiments, fast, but repeat exactly the same way, you should be able to see trend instead of looking for absolute concentration. That's a different ballgame. Whereas if you look at relative, I think we are underselling 2D what it can do. It can do more than we just say, oh, 2D is not quantitative. It is, as you run it exactly the same way. So that's just my comment.
Yeah, good comment.
Yeah.
I have a quick question, if I can. So you showed in the NMR spectrum, you showed these overlapping peaks, right? And you said it's hard to gain useful information from that. I'm actually struggling with a problem right now that I have to be able to somehow deconvolute peaks and integrate them. And I just thought maybe this is a good place to get maybe your input and other people's input on what are ideas on how to kind of tackle that and resolve that. So I have to be able to integrate those peaks and quantify them, even though some of them are overlapping. So are there ideas?
Yeah, that would be a challenge. That is something I'm working on. One of the advantages I have in these spectra is I do have peaks that are resolved outside of that Michigas overlap. So I can get away from it, but I do have other amines that I am looking at that may be in that issue. So at this moment, I got nothing for you. Hopefully there are some pretty awesome people who can help us out.
Well, with that introduction, I think I can at least give some idea. So I'm Samuel Petrus Korhonen from NMR Solutions. So we are doing computer program that is doing quantum mechanical analysis of NMR spectra. And this is, our software is practically, I think, tailor-made for this kind of, let's say, overlapping low field instruments. So I would invite both of you, actually, if you are interested, then just basically drop me a line via email, and we can see what we can do. So
yeah,
please.
I would like that. Thank you.
Yeah.
Yeah, because we have the high resolution spectra and they are very nicely resolved, right? Clearly, and then when we do run the lower field spectra, we don't.
Yeah, it's a completely different ball game, unfortunately. So
thank you. Yeah. Okay, I will send an email. Yeah, thanks.
Thank you.
Yeah.
Thank you. Jose, did you have a question that you wanted to say?
Petrus' timing is impeccable. I was just wanting to comment on the same. I think if you can get, you can use quantum mechanical analysis. If you can have profiles for individual components, then you can use software like Cosmic Truth to put those profiles together and get a quantitative information. I have one little example on my presentation about that. But yeah, I think that would be a way to go.
Good. Thank you. Thank you.
Thanks.
Well, I propose that we move to Jose's talk, and then we can continue the discussion after all the talks have completed. Thank you, Brenna. Very nice job. So with that, I'm going to introduce Jose Napolitano, where he's a senior scientist at Genentech, early research and development organization. So take it from there, Jose.
Perfect. Well, again, thank you for the invitation, Travis, and thank you to everybody on the IBAN group for keeping these awesome workshops going, not only during the pandemic, but after that. I find it a very useful resource and also a lot of fun. And for my presentation today, I just want to give you just a general overview of ways that we apply Benchtop NMR and other ways that we envision how it can be utilized. Just for background, our group is part of the Early Research and Development Organization. So we take projects from late discovery and we, as an organization, optimize conditions to get a viable process that goes into pharmaceutical manufacturing. So that's our place within the pharmaceutical development. And of course, NMR is one of the main techniques that we utilize to identity and reaction monitoring. So you will see some examples of those during the presentation. I will say that in the last decade, pretty much in every place that I have worked and in every scientific conference that has a Benchtop NMR session, there's always this discussion about what's what's the role of benchtop NMR, and if there is really a place for it in industry. And for sure it is. We can discuss for hours about the advantages of disadvantages of these techniques. I think Brenna gave a very good description of it. So I would just keep it to two aspects of high field and benchtop NMR and that comparison. One is footprint. Not all, there is a lot of action going on in our business that is far away from the magnets, and there is not space or facilities to put a high field magnet in all the places that we would like to have it. Of course, benchtop NMR systems are movable, some of them portable. You don't have to deal with cryogen, so those are certainly advantages. And that's the reason why we have been looking closely into how to implement and how to deploy these systems away from our NMR lab. In terms of performance, it's clear that the high field offers better sensitivity and signal dispersion. There is no doubt about that. But as you will see in the presentation, there are ways that we can work on it. We can go around it. In the example that you see on the slide, this is just this 4-fluoro benzoic acid, and yeah, your signals are going to cover a lot more space, but still there is resolution. You still have the information is still encoded in there, and most importantly, the specificity that both Anca and Brenna mentioned is still there in our low field data. For some of our applications, the limitations in terms of temperature control are an issue for sure. But it's an area in which instrument manufacturers and system manufacturers are working actively, and we look forward to see more progress on that area. So for the first example that I want to show you is very simple. We have been looking into ways to support teams that receive materials that are essential for synthesis. And you might be thinking about studying materials, but one of the often overlooked components of a reaction is the solvent. Most pharmaceutical companies have big chemical warehouses storing large amounts of solvents, and also distribution centers. And as you can imagine, some of those solvents have to make it all the way to the lab to be analyzed. So why not put in an NMR, a small benchtop NMR in the warehouse? That's something that we have been looking very actively and in collaboration with AbbVie, another pharmaceutical company. And the idea is just to do very quick identity tests on solvents neat. Benchtop NMR is a perfectly valid way to do it, and it has some advantages over techniques that are commonly used for this purpose, IR, Raman. As you can see in these 6 examples over here, specificity is there. There are clear signal patterns that we can follow and associate with different solvents. If you want more information on this and you are attending PANIC, there will be a short presentation during the Benchtop NMR program at PANIC, and I look forward to see you there. Of course, there is always the critique that you get very often is, well, you can only use these for very tiny molecules like these solvents. And in the case of the chemical warehouse, this is exactly what we're interested in. So it's perfectly good for us. But what if you are interested in analyzing something that is a little more complex? And now we're thinking about how can we deploy NMR, again, away from our NMR lab and more into an analytical lab or a compendial lab in which they are receiving materials, sometimes internally developed, sometimes materials that are coming from contract research or contract manufacturing organizations. And again, there is always some lag in time on receiving samples, sending them to the NMR, and being able to release these materials. So the material that you see in the screen is a little more complicated, and the question is, can we make any sense of this pattern? And as Petrus said, there are strategies that we can use for it. In this case, we use quantum mechanical spectral analysis to generate NMR fingerprints for our products. To get started, you will need the NMR spectrum and a molecular structure, a 3D model of the molecular structure, so that you can use the software to calculate an initial spectrum. That spectrum is within the software optimized until it matches the pattern that you see on your low field data. What you get out of it is, atomic assignments, chemical shift, coupling constants, and integrals. So as you can imagine, if you have a new product, you go through this process, you can generate a fingerprint that matches the pattern that you observe on your Benchtop NMR data. And then if that new lots of that same product are coming your way, you can now use the fingerprint that is just a digital profile that you can attach to a new analysis to compare and make sure that the pattern matches the expected spectrum. In our case, also, we are fortunate to have high field instrumentation, and some of the materials that we have seen before in our lab. So we can modify slightly this workflow to take advantage of the higher signal dispersion of the high field NMR. So in this case, if we have a high field data, we can generate that fingerprint using the high field data and then feed it into the Benchtop NMR data. Remember that parameters like chemical shift and coupling constants are field independent. So it's very easy using modern software to apply this kind of process to mimic the Benchtop NMR pattern and explain what those complex signals really mean. Just to give you a quick view of how this looks in the actual software, as I said, you need the molecular structure to get started. And here in blue and red, you can see the experimental and simulated NMR data and also the residuals. Something that I really like is there is some consistency analysis. I can tell you in the fly something is out of place. If your couplings are not matching or your chemical shift are not matching, that's something might be out of place. Also, if you have other components that are contributing to the overall intensity, that's going to affect the intensity of the overall spectrum. And so you can use as part of your control of the analysis. Of course, this is a more visual part of the analysis, what we see in the screen, but you can also take advantage of that digital profile to have more handles, more descriptors that you can use to verify identity. In this case, on the left side, you have just a comparison between the chemical shift of the same material that I showed you a couple of slides ago. This is just the calculated parameters, both at high field and Benchtop NMR, and they are a very tight match with the average chemical shift difference below 0.01 ppm. Same for the couplings, and difference are below 0.2 hertz. And this really, knowing that all the couplings that we see and all the chemical shift that we can extract from this data match, increase our confidence on the proof of molecular structure. So yeah, just as in the questions that we had right before the presentation, there are ways to use computational strategies, in this case, the quantum mechanical spectral analysis, to go after these signals. One last application that I would like to share with you, of course, is reaction monitoring, just analysis of dynamic processes using time course data. In our case, of course, since we have to optimize chemical processes before we move into manufacturing, there is an interest by our process chemists to have options to run the reactions and not necessarily attached to our high field instruments. So you can imagine deployment of low field instrumentation in analytical labs, synthetic labs, going all the way up to a GMP suite, a kilo lab, a pilot plant, and into manufacturing. And I think there are a few very interesting publications out there on how low field NMR has been applied all the way to manufacturing. The example that I'm showing the screen is one of my favorite go-to molecules when we're setting up new equipment. This is just a chlorine source, dichlorodimethyl and as you know, when you dissolve it in THF, it slowly releases chlorine, and you can see in this case the intermediate being formed, and then how the final chlorodimethyl is formed. Just another example before I go to the conclusions. It's something more process relevant to us. In this case, is the formation of aryl magnesium chloride using going from the aryl bromide and turbo Grignard. And what we're doing here is just helping the chemists understand how long this reaction takes, but also how stable that aryl magnesium chloride is. How long can I hold it? Do I have to use it right away? Can I telescope it to the next step of the reaction? And as you can see, in this case, we have two handles that we can use. We can rely on the hydrogen data, or we can also use the fluorine atoms to follow this reaction. And as you can see, in both cases, the hydrogen and fluorine data are highly comparable. So, in conclusion, I hope that with this short presentation, I give you an idea of how the Benchtop NMR system can be deployed away from our traditional NMR settings, and how they can have a very important impact on how we do pharmaceutical development. We have efficient workflows for structure verification and reaction monitoring in place. And also, as you saw during the presentation, advanced software solutions that helps you analyze complex data set and automate processes are very important. I want to thank everybody that has been involved in one way or another on the information that you saw during this presentation. Our team, the SCG and the Structural Elucidation Group and Process Analytical, our colleagues at AbbVie and University of British Columbia, and then also instrument manufacturers and software developers that they are an important part of our operation as well. With that, I'll like to thank you for your attention, and I'll be happy to take any questions you may have.
Very nice, Jose. Thank you. Very nice. Yuman, do you want to read your question in the chat?
Hi, Jose. I'm just curious about the Grignard reagent degradation step, right? So, for that step, is your Grignard reagent just stored in a sealed NMR tube without any kind of exposure, or yeah?
So yeah, when we start these reactions, we do it several ways. The first test runs that we do tend to be sealed tubes or J-on tubes, in which you can prepare the mixture of the reaction in a glove box. To be honest, in most of these reactions, we like to try a wide variety of conditions. And especially for a reaction like this, we also will try it in flow, in which that the solvent, the reaction mixture is going to be, through the headspace, is going to be exposed to more air and moisture. And that way we can also have an idea of what's the worst case scenario. But yeah, okay. It's depending on the specific process, and if you are dealing with a homogeneous mixture or not, we use a combination of static, just NMR tube analysis and flow analysis.
Yeah, cool. Thank you very much. Because I'm asking because I have some previous work characterizing the stability of similar granular reagents, right? But our finding that for the granular reagent itself, if it's like a well sealed, even in air like room temperature, it wouldn't degrade like so much. So that's why I was like curious to ask about it. Yeah, thank you very much.
No problem.
Was that in a co-solvent, Jose? The analysis for the Grignard, or was that neat?
For the Grignard, I think if I remember correctly, it's in THF.
Okay.
Yeah.
Okay.
I think we have THF in there. Yeah. So it was performing THF at room temperature.
So what, about 10% solids or ballpark?
I think it will be around 10, probably 12, no higher than 20%.
Okay. Okay. Thank you. Any other questions?
I have a quick question that might not be truly applicable, but when you're doing those comparisons of your data against the predictions of the spectra from chemical structure, so for benchtop, the sensitivity is an issue, right? So when you have that sample limited, that sample, that chemical that you're interested in, and it's limited in terms of the amount that you can get, how do you work around that? Like, are there any other strategies that you would use to kind of circumvent that lack of sensitivity on the benchtop, apart from going to a bigger magnet, of course?
I'll be honest. In our case, because of the stage that we are in the process, we are not sample limited most of the time. Our first clue always, if you are dealing with signal to noise problems, is collect data longer, which it doesn't help if you don't have a lot of material. In the case of the calculations, I would say it's a factor, especially if you have other intense peaks in there that might be not related to the structure. I would say that in the case of the examples that we have done, the software is, as you put your molecule in and your spectra in and you define the solvent, the software is taking care of at least your solvent peaks, your water peaks. So that means that when you're trying to reduce your root mean square to get into explain most of the intensity of the spectrum, the focus is going to be on the analyte itself. But yeah, if you have more components in there, you are going to have, it's going to be more difficult to have a really tight match in terms of intensity, especially for low level impurities. If you have patterns for all of them, you might start being able to go down on the intensity scale. But yeah, I really don't have a good answer for very sample limited material analysis.
Perfect, thank you.
Hello, could I jump in? I think I have a related question. So you mentioned that the prediction can handle the solvent. I was wondering what happens when you have things like NHs and OHs coming from your molecule, and that they're notorious in predictions for when you want to compare against NMR samples, because they're concentration dependent, pH dependent, temperature dependent on absolutely everything wandering around your spectrum and getting in the way. So I was just wondering how it handles those.
Well, I will say that the first thing, and maybe it wasn't, the presentation didn't went too much detail into it, is that it's not only a prediction. Once you do the prediction, you can also, you have a graphical interface in which you can actually help correct some of those predicted values until what you get in the end as a fingerprint is not really only a prediction, but an optimization on the prediction. So yeah, OHs and NHs are going to be notorious to be off of the predictions, but you can manually account for those. And assuming that your samples, if you are working on lot to lots and the pH or salt contents on those samples are not substantially different, the fingerprints are going to be able to account for those. But yeah, it's more than just a prediction. The final pattern that you get from the fingerprint allows you to, the process, the iterative process to get to that point allows you to account for those differences.
Brilliant. That exactly is what I was hoping to find out. Thanks.
No problem.
Good question, Claire. Thank you. Anyone else? Specifically for Jose? All right. Before we open it up, I just want to thank Anca, Brenna, and Jose one last time for very good presentations, very interesting, and thank you. Dave, did you want to say something?
So are there still questions?
I don't think so at this time.
Oh, okay, great. So I would like to thank everyone for coming, and this has been a very interesting meeting with a lot of applications I wouldn't have thought about. And so thanks everyone for coming. And with that, I'll close the meeting.
Great.
By the way, we always have this nifty Ivan gavel. So let me.
Thanks. Thank you, everyone.
Thank you. Thank you, everyone.
Very nice.